Hi, everyone. Welcome. Thank you for signing on. Right on time. Thanks very much. Welcome. My name is Haley Ross, and I'm here as your host. I work with the Columbia Mountains Institute of Applied Ecology, and we are here to welcome you today for the 6th season of CREDtalks, the Columbia Region Ecological Discussions. This is our 3rd talk out of an 8-part series. And the CREDtalks this year will explore the theme of climate disruption in the Upper Columbia Basin. We have 8 scientists who are going to address projected impacts, potential for adaptation, and what we can do with ecosystems that may in fact contribute to mitigation. If you're in attendance today, I'm sure you've already seen the impressive list of speakers that we have in the series, but if you're listening to the recording, I encourage you to go to the event webpage. Excuse me, I'm stumbling over my words. I encourage you to go to the event webpage to see the full suite of speakers in the series. And you can find that at www.cmiae.org. You should see the address on the top right-hand side of your screen. Today we're going to welcome Dr. Janice Brahney, who I'll introduce in more detail in just a moment. But before I do, I'd like to thank the Columbia Basin Trust. The CREDtalks are financially supported by the CBT, and we share our gratitude with them for providing the funds that we needed to get this project up and off the ground. The series is also generously supported by all of you. Without your donations, this project would not be possible. So thank you very much for your support. It's very much appreciated. And we also thank everyone viewing the recordings and sharing this resource with your network. If you do review this at a later date and it's possible to donate, you can do that via the event webpage, as I already indicated, and thanks to you for that as well. Before going any further, I'd like to just stop for a moment and acknowledge and honor the 4 nations on whose unceded territory I broadcast from today in Revelstoke, BC. So that is the Sinixt, the Ktunaxa, the Secwepemc, and the Silks. And what I'll do now is I'll actually welcome all of you to introduce yourself, and please feel free to let us know whose territory you're zooming in from today. So to do that, we've enabled the chat, and if you're unfamiliar with Zoom, you'll find the chat bottom on the bottom control panel of your screen. So if you hover your mouse down there and you open the chat, then feel welcome to, like I said, introduce yourself and give a little land acknowledgement. In the spirit of reconciliation. So thanks. I can see that we've got people on board who have been through the first 2 talks already. You know the routine, and those acknowledgements are coming in. Thanks very much. I'm going to take a moment while you're introducing yourselves to talk just very briefly about CMI, who we are and what we do. And I know that many of you have heard this introduction a few times now, and if you stick with us for the full series, you'll hear it a few more times. So thanks for your patience as I run through this. So the Columbia Mountains Institute is CMI, what we call ourselves for short, and we're a nonprofit society or an association of people working for people who work in applied ecology. Our home range is southern British Columbia, Canada, and increasingly we see membership from across BC and into Alberta, the Yukon, and the Northwest Territories. One of the main things we do is provide professional development opportunities. Those typically come in the form of conferences, courses, and webinars. Now, typically I run through a bunch of examples of what this looks like, but in the interest of time and in the interest of those of you who have heard this intro many times, I'm going to skip through that and just refer you to the website for more information. So again, cmiae.org has a huge list under our past events of all of the types of events that we typically hold. And there are also really great resources on there. So for example, our large events have proceeding documents where you can learn more about the resources and the debates and that sort of information that was shared at those events. Our audience is typical audience, I should say, is actually well captured by the registration for the CREDtalks. So when I'm going through that registration, I'm seeing representatives from environmental consultants, academics, students, industry representatives, nonprofits, First Nations governments, and provincial and municipal resource managers. So thanks for your interest, and again, welcome. So now what I'm going to do is run through some nitty-gritty details, a little bit of a Zoom orientation, a little bit of info on how the webinar is structured today. First off, I'd like to recommend that you take in the webinar using the speaker view. So if you hover your mouse on the top right-hand corner of your screen, you should see the little view icon pop up. And if you click on that, you can experiment and see what you like best, but I'm going to recommend speaker view. I'd also like to let you know that the webinar is being recorded and that the recordings from these webinars are available on the event webpage. So the prior 2 talks and all of the talks to come will be posted there within about 1 week of the talk having taken place. We're in the webinar format, of course, and this means that attendees have had their cameras turned off and you've been muted. If you would like to be able to speak, however, like for example, if you would like to ask your question orally once we get to the Q&A instead of typing it onto your screen, you can use your raised hand function, and we'll take that as a cue to unmute you, so to speak. So again, you find the raised hand icon at the bottom of your screen in your control panel. So I just put mine up. You probably see a little yellow hand next to my face there, and I can lower it as well. The chat feature is enabled, as you saw, and this is enabled so that we can do what we just did, introducing and sharing our acknowledgements. It's also there if you would like to share resources with one another. We're not going to use the chat for the Q&A, however. So for questions, what I'd like to do is just direct you to the Q&A feature. Again, that's an icon at the bottom of your control panel, the bottom of your screen on the control panel. If you click that open, you'll see another box open up, and that's the Q&A function. And we're going to be using that for our questions. You are welcome to type in questions into the Q&A as they come to you, but we're going to be holding questions until the end. I think Marc-Andre is probably in the audience, or one of the CMI directors. If you could just pop an example question into the Q&A for me for a moment. I want to just demonstrate the upvote feature, which we're going to be using. So if somebody types a question into the Q&A and you think, oh, that's a great question, I want to hear an answer to that. Once the question appears, I don't see one yet, but there's hopefully is going to be a question appearing any moment. There will be a thumbs up icon that appears right under it, and you can click that thumbs up. If there are a number of thumbs ups, I guess, that appear with that question, it'll pop up to the top, and then I will ask that question first. So great, thanks. We've got a couple of questions there, thanks. And you can see that Marc-Andre's question is quite a bit more popular, or was for a moment, than Brendan's, and it came up to the top. So I'll be relaying those questions at the end in the order of popularity. Thanks. All right, I think that's about it for all of the nitty-gritty details, and now we can launch into the reason why you're all here today. So it's my pleasure to introduce Dr. Janice Brahney, who will be presenting climate change impacts on water quality in the Columbia River Basin of Canada. Dr. Janice Brahney is an associate professor at Utah State University. She received her doctorate from the University of Colorado, Boulder, in environmental biochemistry. She holds her MSc degree in the Earth Sciences and a BSc degree in environmental science, both from Simon Fraser University, Canada, of course. Her research sits at the intersection of several critical zone disciplines and includes 3 primary themes. 1, the atmosphere as a vector for material transport to aquatic ecosystems. 2, climate change effects in mountain environments. And 3, the cause, effect, and mitigation of water quality impairment. Dr. Brahney is currently an associate editor for Freshwater Science. And we're really grateful to have her here today and lucky at that. So Janice, I'm going to pass it over to you. Thanks very much.
Hello, everyone. Oops. Thank you so much for inviting me to talk today. I am speaking to you today from Utah on the traditional lands of the Ute and the Shoshone. So I have been working on climate change impacts and water quality for some time now, and much of that work has taken place in the Columbia River Basin of Canada. So that's what I'm going to be talking to you about today. I watched the previous talks, and Brian mentioned that while he was a student in Colorado, he worked with Mel Reisner on mountain lakes. And so I wanted to continue that chain and let you know that when I was a student at Simon Fraser University, I worked with Brian. He was one of my advisors. And we did a project trying to understand how glacial fluctuations influence Kluane Lake levels. The only photographic evidence I could find is this, Brian's elbow, but I promise you that he was my advisor. Okay, so on to the topic of today. Climate warming has the potential to influence freshwater ecosystems through numerous different pathways, but today I'm going to focus on just two pathways. The first is increases in wildfire, and the second is the loss of glacier resources. Both of these have the potential to change the physical and chemical environment of freshwater systems, which in turn can lead to a community reorganization. So if you're changing the habitat, you're going to change who lives there. And in addition, we often talk about emergent consequences of these types of changes. And this can be attributes that change that are difficult to predict. And so I'm going to talk a little bit about that as well. So I like to call this talk a song of ice and fire because I'm a big fan of Game of Thrones, and we're going to start with fire. So as you've heard in the previous talks, we're transitioning into a drier and warmer climate. And we all experienced a much warmer climate last summer. And this can lead to increases in the incidence of wildfire. Over the past couple of decades in the southern United States, we've definitely seen an increase in wildfire, and that can impact British Columbia, and I'm going to speak about that a little bit as well. So the way that fires and wildfires can influence aquatic ecosystems is through direct mechanisms and through indirect mechanisms. The direct mechanisms are the really obvious ones. So if you have a fire burning in a catchment, it's going to remove the vegetation, it's going to add soot and ash to the aquatic ecosystems, and that can result in these very substantive effects. And I'm going to show you some data describing that in detail. But there can also be these indirect effects, and this is related to this heavy smoke cover that can occur and persist for long periods of time. The role of smoke can impact areas that are 1,000 times greater than the actual area of the wildfire. But we have a lot less information on how this indirect effect can influence water bodies through dispersed water bodies. But we can hypothesize that it has something to do with changes in the light environment as well as changes in nutrient deposition. So I'm going to start by talking about the direct effects of wildfire on aquatic systems using a case study that took place in Okanagan Mountain Park. This was work that my postdoc advisor, Jeff Curtis, did in the Okanagan. In 2003, there was a large fire in Okanagan Mountain Park that completely burned a large area. And so this created the opportunity to do a paired watershed study. And what we did was compare areas that were completely burned, so watersheds that lost all of their vegetation, to very similar watersheds that were not burned. And so these were similar in area, similar in aspects, similar in elevation. So we could directly compare these two types of watersheds. So looking at some of the data here, in red is the burned catchment and green is the unburned catchment. So this is showing yield, so the amount of water that moved through the system. And you can see that in the burned catchment, immediately there was much larger water moving through the system, and that peak happened much earlier. So the reason we see a lot more water flowing through a greater yield in a burned catchment is for two reasons. One, all the vegetation is gone, so there's less evapotranspiration, less water being taken up by the trees and being transpired to the atmosphere. but also because fire can really substantively change the soil environment, change the structure and its ability to infiltrate water. So it increases the amount of overland flow that occurs. And that can lead to really substantive erosion and infilling of and sediment transport. So on the upper right, you can see a water body that has been infilled by a lot of sediment that has been eroded from the catchment. And on the bottom right, you can see a river that is very turbid and undergoing active erosion because of the high flows that have occurred post-fire. So obviously this changes the habitat environment quite a bit and can be really detrimental to fish, not just because of the high turbidity, but because the ash and the high organic content of the sediments in the water can consume the oxygen that is present, leading to hypoxia and fish kills through that mechanism. If we look at the temperature, you can see that the burned catchment was on average 2 degrees warmer than the unburned catchment throughout the summer season. And this data is from 1 year post-fire. And so you might ask, how long would we potentially see these types of impacts? Well, a 7-year study that took place just south of the border in Montana showed that this 1 to 2 degrees increase in temperature persisted for the full 7 years of that study. And the degree of increase in temperature was correlated to the intensity of the burn. And the reason the temperature is increasing is because all of that riparian vegetation has been burned. And so there's higher rates, higher insulation on the water. So it's warming much easier. So we might anticipate that it could take up to a decade or longer for this type of impact to be mitigated because we have to wait for the vegetation to grow back. But during that period of warmer waters, that can lead to issues for the endemic cold water fish species that are adapted to colder temperatures, and it can also promote the invasion of warmer water species. Next, if we look at the amount of phosphorus within the burned catchment, we can see that a much greater amount of phosphorus is being moved through the system. We have much higher phosphorus concentrations. And phosphorus is a really important nutrient. And so that can sound like a good thing, a nutrient. But since phosphorus is typically very limiting in ecosystems, we don't want an increase in phosphorus. Algae are adapted to having very low phosphorus in their environment, and so they have a lot of different mechanisms to acquire phosphorus. And when it becomes freely available in higher concentrations, that can lead to the explosion of algae in these algal blooms that can foul ecosystems. It can lead to the excessive production of organic matter and the consumption of oxygen, as well as the production of toxins. So in general, we don't like to see higher phosphorus concentrations. A review study that was published in 2011 showed that post-fire fluxes of phosphorus can be elevated by up to 430 times, and post-fire fluxes of nitrogen, another important nutrient, can be elevated by up to 250 times. So again, we might ask, how long will these impacts persist in burned catchments? Well, there hasn't been a study long enough to determine exactly how long these impacts can persist. But a 5-year study in the Central Rockies in Colorado showed that nutrient concentrations remained elevated through the full 5 years. But what they did start to see through that time period is that catchment slope could potentially control the rate at which watershed recovered. So really steep catchments seem to have a faster rate of recovery than low slope catchments. And this is likely because the steeper catchments have a greater capacity to remove the material, the ash that has accumulated in the catchment. And then finally, looking at concentrations of dissolved organic carbon, we actually see that these are lower in the burned catchment. And this makes a lot of sense. A lot of the organic carbon is coming from the vegetation that was burned and turned into carbon dioxide, and because of the changes that happen in the soil environment. So normally when we think about DOC and water quality, we don't want a lot of DOC because it changes the color and the taste, and it can impact our ability to um, treat water for drinking. But in these high mountain environments, we actually want DOC because it absorbs harmful UV radiation that would otherwise act to suppress biological activity. Okay, so these are some of the really direct effects that occur in a catchment that has had a fire. But what about some of these indirect effects? So this is an image from last summer showing a high smoke, medium smoke, and light smoke cover over the continent in North America. For much of the summer, large areas of the continent, in particular the West, were covered by smoke. And so this can lead to an increase in nutrient deposition because ash can be up to 50 times increased in phosphorus and nitrogen in nutrients compared to the vegetation it came from. So ash is often really enriched in these nutrients. So it's being mobilized into the atmosphere and then being deposited widely across the ecosystem. Smoke also decreases the amount of light that's being transmitted to ecosystems, so it has the potential to influence primary production. So if we look at all of the different potential elements that can be volatilized when you burn vegetation, you can see in this image here, there's a lot of different nutrients, not just nitrogen and phosphorus, but also calcium and magnesium and potassium. So there's this potential to move those nutrients into the atmosphere that can get deposited. But you also see that mercury is listed here. So there's also some toxic elements, toxic heavy metals that can be volatilized and transported long distances. We know that from areas that have high rates of biomass burning on purpose. It's part of the landscape management strategies in certain areas. We know that this heavy burning of vegetation can move so many high amount of nutrients through the atmosphere, so much so that nearby water bodies have become really enriched in nutrients to the point of detriment. So we know that that's a potential mechanism. But there hasn't been a lot of work trying to understand what's happening in North America or in Western North America on the role of fires and nutrient transport through that pathway. But what we have done in the last couple of years is try to quantify this. And what we've seen is that when there's smoke in the atmosphere, we can see up to 40 times increase in the amount of phosphorus that's being deposited to ecosystems. So we are seeing that increase in nutrient deposition. And last year, a study was published that looked at smoke cover over a lake system and the effect on the nutrient concentrations. And so this is a lake that has many years of research. It's got many in situ sondes measuring all kinds of things, and it's got a research station. So there's people constantly collecting data. So there's a really excellent data set. The blue line represents the mean and the distribution of nitrogen concentrations through the last 10 years or so, whereas the red line is representative of that heavy smoke year. And you can see that there was a really significant increase in the amount of nutrients within this lake system that occurred during that smoke cover period. So this is actually data from Castle Lake in California, which in 2018 had 55 days of smoke cover. There was a fire burning in a different catchment, but it was close by. And so you can see the image of how heavy the smoke was. It was quite intense for certain periods of time. And that also led to changes in the light environment. So on the left, you can see that there was a really substantive decrease in the amount of harmful UV radiation. And on the right, you can see there was also a decrease in the amount of PAR, or what we call photosynthetically active radiation. So the wavelengths of light that are used by organisms in primary production. So despite these decreases in the light environment, what they observed was an increase in primary production. So they saw that productivity increased and moved to higher in the water column. And so this might be due to a combination of that release of harmful UV radiation as well as nutrient deposition. They also noted shifts in the zooplankton community composition through this time as compared to other years, and modifications in fish behavior. So this is just one lake. As I mentioned, this is the effect of smoke on aquatic ecosystems is still not very well understood. We don't have a lot of data, or not a lot of research has occurred to date. But as I mentioned at the start, smoke can impact an area that is 1,000 times larger than the actual fire. So it is something that we should try to understand. Okay, so moving on to ice. Brian gave us a great talk on how we are losing our glaciers, and we know already that in the past glaciers have receded quite substantively, as you see on the left here, this image of the Illecillewaet Glacier. And the projection is in the future that we are going to lose our glaciers entirely. So what does that mean for the ecosystems that typically receive glacial water? Well, that's a difficult question to answer because we can't conduct an experiment or use an event like a fire to try to understand that implication. So how do we build a roadmap to the future on what we are to expect? So one way that we can do this type of study is called a space for time substitution. So I'm going to illustrate that concept using an elevation gradient. So on the left, we have a mountain environment with 3 different types of vegetation, a lower elevation, mid elevation, and high elevation ecosystem. And in the future, it's anticipated to change. And if we want to understand what that mid elevation system is going to be like in the future, we might study the lower elevation ecosystem to get an idea of what that might look like. So that's what we did in the Columbia Basin of Canada, is we did a space for time substitution to try to understand how our rivers and lakes are going to change in the future under a different glacial regime. And so we sampled, as you can see in this previous slide, many different sites around the basin, and we classified them into sites that are actively glaciarized. So those are sites that have at least 5% glacier cover in the catchment. So you can see in this image of Joker Lake on the left that the red polygons are glacier cover from 1985. And so you can already see there's been some decrease in glacier cover at the headwaters. And we sampled their rivers that are down valley. So this is the Illecillewaet, not coming off the Joker, but it's a nice example of a heavily glaciated system. You can see that turbidity from the glacier. Our second category we called transitional systems, and these are sites that had less than 5% glacier cover. And we sampled the lakes in these catchments as well as the down valley rivers. And the transitional sites also included areas that had recently lost glaciers, so in the last couple of decades, but still retained some glacial character, so high turbidity systems. And then our final category were snow-fed systems. So these are sites that had not been glaciated at all in recent history. And sometimes we separated these into bare sites, so rocky catchments versus forested sites, because that can influence the water chemistry and the ecology. And the great thing about the Columbia Basin is sometimes we can get all of these sites in one single catchment. Okay, so I'm going to show a lot of data, and I'm going to use these 2 different icons when I'm talking about lake data versus when I'm talking about stream data, just so that you guys can orient yourself to the data on the slides. So in terms of the loss of glaciers and the impacts on lakes, it's going to have a really significant effect on the physical environment. And you can see that visually here with this shift from these beautiful turquoise colors that are due to the high turbidity from the glacial flow to these deeper, darker blue colors that are representative of more clearer lakes. And so with that shift, we can expect a change in the light environment. So under really turbid conditions, we have lots of particles in the water body to absorb both the UV radiation that can be damaging, as well as the light wavelengths that are used for primary production. So in this particular graph, the damaging rays are shown in purple, and the rays that are used for photosynthesis are shown in green. So in turbid environments, they're both attenuated very rapidly, whereas under clear conditions, they can both penetrate quite deeply into the water column. And then once we start to have vegetation encroachment on the environment, there can be more dissolved organic carbon entering the system, which can absorb the harmful UV radiations that I talked about a little bit before, but allow the photosynthetically active radiation to penetrate much deeper. So based on these changes, we can anticipate seeing an increase in production as we lose glaciers from these systems. So that's a function of the changing light environment, as well as the longer growing seasons that are facilitated through warmer temperatures, as well as the warming of the water itself, which can lead to more production. And so in our three different categories, we do see an increase in the amount of organic matter in the system, in the sediments, as a result of that increased production as glaciers are lost from the system. We also see an increase in temperature as predicted. So these are mean summer temperatures. You can see they're quite a bit higher in the absence of a glacier. And with higher temperatures and longer growing seasons, we might anticipate also seeing more evaporative loss from these lakes. And that's in fact what we do see. So you can see that the non-glacier ice systems have more than twice the amount of evaporation to inflow as the glacier ice system. So we can expect that not only will we see loss of water from the encroachment of vegetation, but also from the water surface itself. So when thinking about temperature, that's not only important for these physical environments, but also an important habitat control. So on the upper left, we've got monthly data from our three different categories of glacierized, transitional, and no glacier. And you can see the glacierized systems are quite a bit colder. And on the bottom is for our lake systems. But we also know that our glacierized systems are higher in elevation, and that's going to have an impact on our temperature. So if we look at the relationship between air temperature and water temperature, we can get a better sense of what the glacial suppression of temperature is in those particular systems. And you can see in the river ecosystems that in the summer and in the late summer is when we really start to see that glacier effect in suppressing those water temperatures. So the effect is about a 3 degrees suppression in temperature. And in the lake systems, it's almost 6 degrees. Now, another way to think about the distribution of temperature and the effect of glaciers is to look at the distribution. So here on the y-axis, we've got the frequency, so the amount of time that each of these systems are spending at a given temperature. So you can see in June and July, there's this nice separation between glacierized, transitional, and snow-fed systems. And that separation increases in August and October, where the glacierized systems are more distinct and the transitional and snow-fed are a lot more similar. And this is an important time of year because this is the time of year when a lot of fish are moving from lakes into river systems to spawn. So if we consider the ideal spawning temperature of some fish, say between 4 and 14 degrees Celsius, you can see that the glacierized systems encompass those temperatures very nicely and almost never go above those temperatures. Whereas as we move into the transitional and the snow-fed systems, these river systems are spending an increasing amount of time at these temperatures that are non-ideal or even lethal to some fish species. Okay, so that was the physical environment changes. Now let's talk about the chemical changes and the biological changes. Across our three different classifications, we see these really large changes in the chemical environment. So we have a much higher conductivity with the amount of glacier flower contribution, as well as much higher nutrient concentrations. Glacial systems have higher nitrate in particular, as well as phosphorus, and much greater turbidity. In opposite, we see as the vegetation starts to encroach on the landscape, we see increasing dissolved organic carbon concentrations. So this is what we might expect to see. And if we look at the community composition that's present, we see these shifts that are associated with these physical and chemical changes that are occurring. So on the left, we have a graph that is representative of all the different community data that we collected from these different lakes, collapsed into just two different axes. And the way to interpret this type of graph is that each dot represents a unique site. The closer those dots are together, the more similar the species compositions are, and the more further apart, the more different they are. So you can see that our four different categories are grouping very nicely and are distinct from each other. So our glacierized community is different than our transitional community, and so on. On the right is showing a distribution of those species through our four different classifications. And here I separated the snow-fed from the snow forest systems so that we could look at those differences. Now, what this is translating to is difference in the habitat uses by different species. So in our heavily glaciarized systems, we dominantly see organisms that live attached to rock or epilithic species, because in these systems, they have very rocky bottoms and high turbidity, so there's not a lot of light penetration. So these organisms tend to live in the shallower areas on the rocks. But as we moved into the transitional systems, we start to see organisms that live in sediments, in sand, and in organic sediments. As we move into the snow-fed systems and we start to see a little bit more of that dissolved organic carbon entering the system that can absorb that UV radiation, we start to see planktonic organisms appear. And then finally, in the forested systems, we start to see more vegetation in the nearshore environments that create a new habitat, and we start to see organisms that live on other vegetated species. So with the loss of glaciers from a system, we can anticipate seeing coincident changes in diversity. So what I just demonstrated was this red line here, and this is from the Milner paper in 2017, which is making predictions about what might happen to diversity as glaciers are lost. And we essentially saw this, that as we increase the amount of diversity within a given ecosystem, we see we increase the diversity of habitat, we increase the diversity of species that are present and using those habitats. But the anticipation is that we're going to see a decrease in regional diversity, and that's because we're going to lose these highly specialized taxa that are adapted to these cold glacial environments. And looking at our diversity across the systems, that's essentially what we're seeing. So in our glaciarized environments, we see very unique species that are highly adapted to these high turbid environments, high turbid and cold environments. And then as we move into the transitional, we see an increase in diversity because we're seeing some of those highly adapted species still present, but we're also starting to see new species enter that are more adapted to warmer climates. And then as we move into the rock and forested systems, we lose those highly adapted, unique species that are present in the glacial system. So we see an overall loss in that regional diversity. Okay, so when we think about now moving into the impacts for stream ecosystems, we have an additional dimension to consider. So we're not just changing the temperature and the chemistry, but we're changing the timing. So on the left is the long-term decline in glacial melt contributions to, in this case, an example, the Illecilla River through time. But what I'm showing on the right is what's critical is that we're losing that water in a specific time of year. And so that's in the midsummer to late summer period. And so this can lead to really substantive changes in the chemistry through time. In particular, for turbidity and phosphorus, we see this major shift in the timing. So for both of these two parameters, you can see a very large peak in the mid to late summer. But as soon as we move into those transitional systems, that peak is much reduced, but it shifts to earlier in the time of year. So not only is it smaller, it's happening at a different time of year. And we see that both with turbidity and with total phosphorus. With dissolved organic carbon, we see an increase in the amount of DOC with each different category, as we might anticipate with more vegetation. And the peak is only slightly shifting back in time. So if we look at the diatom community that lives in, or the periphyton community that lives in the river systems, in that same way we looked at the lake communities, you can see that there is some difference between the communities and our different classifications. But what was really surprising to us when we were doing this field work is that we saw Didymosphenia geminata only in non-glaciated rivers. And so if you've not heard of dynamo, what is dynamo? Well, this is a benthic diatom that in recent decades has been exploding in terms of being more dominant in the landscape and leading to excessive overgrowths. And this was actually first noted in Victoria by Max Bothwell and his colleagues, where they noticed this sudden explosion of this particular diatom species. So even though they're microscopic in size, these are their cells that are only about 100 microns long, so really tiny, and they attach to the rock substrate with these stalk. And the interesting thing about this particular organism is that it can produce a lot of stalk. And so that's what you're looking at the right, is all of this stalk. The cells are at the surface, and all of this is just stalk material that attaches the organism to the rock. And so this can lead to really excessive fouling of water bodies. And as I mentioned, this has been noted in many places around the world in the last couple of decades with increasing frequency. And so there's a lot of questions as to why this is happening. Initially, it was hypothesized that this was this was an invasive species and it was being moved to different river systems by fishermen. But we've since learned that this particular organism is endemic to North America. It's found all over northern latitudes, actually, and it's only just recently started to change its behavior, if you will. So there's a couple of really interesting things about this particular organism. So as we were talking about earlier, when you add phosphorus to an ecosystem, a lot of different algal species will explode. So they'll start to divide their cells and you can have a massive algal bloom. With diatoms, we see that same relationship. As you add dissolved reactive phosphorus, which is what DRP stands for, is you increase the amount of phosphorus in the water, you increase the rate at which cells are dividing. So FDC stands for frequency of dividing cells. So that's this component here. You can see in this case, there's two different cells that have divided. But with this particular organism, there's an inverse relationship to the frequency of dividing cells and the amount of stalk that is produced here using the index, standing crop index. What that's referring to is the amount of stalk that's being produced. So that means that under low phosphorus conditions, we tend to see this organism start to produce a lot of stalk and foul the environment. The other thing that's been noticed about this particular species is that under high light, there tends to be more of that standing crop. There tends to be more of that biomass present. And so this led Max Bothwell to hypothesize that what's happening, called photosynthetic overflow. And this is because there's a high light environment, so there's a lot of energy for photosynthesis to occur, but there's not enough nutrients for cell replication to occur. So all of that energy that's being produced and utilized to convert into carbohydrate is getting shunted into stock production. So that's that overflow component. So instead of dividing cells and replicating, all of that energy is just going into producing a lot of stock. Okay, so why is this happening more recently? In general, we've increased the amount of phosphorus on the surface landscape. So this seems in opposition to how we've been affecting our ecosystems broadly. But if you recall, we are changing the timing at which things are happening. So what's shown here is the photoperiod in red. So that's the amount of light in a given day throughout the year. That's not going to change with climate change. That's related to the tilt of the Earth. And in blue is that turbidity and nutrient peak that we see as a result of glacial melt as well as the freshet. And since that is moving earlier in time, we no longer see an overlap of that turbidity and nutrient peak with peak photoperiod. So what we are essentially doing, or what's essentially happened, is that in the mid to late summer, we have lower flows because that peak is happening earlier and we're losing that glacial contribution. We have increased transparency because we've lost that turbidity. We've decreased the nutrients as the glaciers disappear. They bring those nutrients with them, and the temperatures are increasing. So these four things create ideal conditions for dynamo to start going into that overgrowth life strategy. And so with our initial data set, we wanted to know, are we imagining this? Are we just, noticing dynamo more in non-glaciarized streams? And so we did a bit of a study to try to understand whether or not dynamo was in fact randomly distributed across the environment, or whether or not it was in fact related to glacier cover. And we did find that yes, it was related to glacier cover in our initial study. And you can see that the percent coverage of dynamo increased with the lower glacier cover. But we wanted to do this in a bit more of a controlled way. So we expanded the study. We increased the space for time study. We added an additional 53 sites. We also did some experiments. So we created some experimental flumes where we could test the role of light in enhancing the stock production of dynamo. And so here it's important to point out that glaciers can potentially suppress dynamo blooms through two mechanisms. One is through the light mechanism that we've been talking about in this photosynthetic overflow. And the second is due just to the abrasion from the amount of glacial flour that is in the sediment. And so we wanted to isolate those two different potential processes and just look at the role of light. So we had 3 different flumes and multiple treatments and replicates within each flume. And our treatments were to have a high UV radiation, so no cover. This was to replicate a high elevation system that has no glacier and no vegetation. So there's very rocky catchments that are up there. A forested site, so this is representing a system that has lost a glacier and has some vegetation encroachment, and so there's some DOC in the environment. We also wanted to replicate what the freshet might look like from a snow melt pulse and what glacier melt looks like from the high turbidity. And so we did this by using different covers that can manipulate the amount of UV radiation as well as PAR that is being received by our dynamo. And all of these were seeded with dynamo. So for our expanded space for time study, we looked at how the different physical properties that we're interested in, phosphorus, turbidity, and temperature, predict whether or not we see a bloom or a no bloom. So we created this model where we could look at the probability of whether or not we would see a dynamo bloom or an overgrowth versus no bloom at all. And so it's also important for me to point out that dynamo is everywhere. So just because it's not in this state of creating a lot of stock doesn't mean it's not in the ecosystem. So it's often there, but with many other species of diatom and not taking over the environment. Okay, so what we found is that the probability of a bloom is significantly higher when we don't have a glacier in the system. So in these snow-fed and transitional systems, the probability of a bloom was much higher as related to this extinction coefficient, which is the amount of light which can be passed through the water column, and with the amount of nutrients that are available. So SRP stands for soluble reactive phosphorus. So again, with higher phosphorus concentrations in the glacier ice systems, we see a lower probability of a bloom occurring than we do in these snow-fed transitional systems. The experimental flumes also showed us very similar results to what we were anticipating. So you can see that the y-axis here, this is ash-free dry mass. So this is another metric that is quantifying the amount of biomass that was produced, specifically stock biomass. So all of these different treatments had dynamo present, but what we were measuring was the amount of organic matter that they produced through their stock. So in the glacier melt simulated system, you can see that the lowest amount of stock was produced, and the biomass that was present was largely their cellular biomass. And then in the snow melt and the freshet was also suppressed compared to our forested system, which had the greatest amount of stock being produced. So this aligns with our hypothesis that it's the reduction in glacier melt and a shift to earlier in the time of year for that snow melt peak is what's creating these conditions, allowing dynamo to bloom. Okay, so many of you might be wondering, how does dynamo affect fish? So if we're going to expect to see a lot more dynamo blooms in our river ecosystems, what are the impacts to the fish that we care about? And could this be related to a change in the macroinvertebrate community composition? So macroinvertebrates are important food sources to fish, and they can be really... Here's what I found online. Oh my gosh, sorry. They can be really intensely affected by the amount of organic matter that's on the river bed. And so we wanted to understand how that might impact native fish species. So we conducted a study in the Kootenay Basin at all the different river systems that you see on the upper right. And what we found is that there was, in fact, a shift in the macroinvertebrate communities. Specifically, we saw a decrease in the ephemeroptera, plecoptera, and trichoptera species, the EPT taxa that are typically really good fish food. But on a good note, we saw that it had no impact on fish condition. And this is because these fish are typically not food limited in these systems and can move around. And so even though with increasing dynamo cover, and across our study, we had from 0 to 80% dynamo cover, we did not see any impact on fish condition. So I want to end there on a good note, and I will take questions. Thank you very much for your time, and also I would like to acknowledge the funders of the study.
Thank you so much, Janice. That was great. I appreciate the pace that you went through that for myself anyways. It's so interesting. There's so many connections to illuminate there for us. I don't see any questions in the Q&A at this moment, so I just want to prompt everyone, if you have any questions, again, type them into the Q&A. You'll find that Q&A icon at the bottom of your screen on the control panel to open up that box. And if you see a question that is included there and it's a question that you'd also like to see the answer to, then just like we discussed earlier, give it the thumbs up. And I will read out the questions in order of popularity. Amazingly, Janice, maybe this is working in your favor because I know that you have a meeting in 5 minutes. Our questions at the moment are low, though I'm sure somebody's going to think of something in just a moment. And maybe as their ponder, I'll just say that I appreciated you ending on that positive note. I didn't expect that last slide to say what it said. I thought you were about to illustrate some terrible implications for the fish population.
Yes, no, I wanted to end on something that was good.
Great. Okay, one question here. So Suzanne Iyer, oh, she says you answered her question already. So, oh, here we go. We've got one from Tyne. Do you have any thoughts on the dynamo overgrowth? On how the dynamo overgrowth would affect the riparian or semi-aquatic communities?
Yeah, that's a great question. So we only looked at fish species that are capable of moving. So that if they're in an environment where they don't have a lot of food, they can just move upstream or downstream to where they can access food. But that's not true for organisms like mussels that can't move, and so there could be an impact on those immobile organisms. And you know, I imagine a lot of other species that might depend on food resources or specific habitats that could be influenced.
Okay, great. Here we have a comment, possibly a question from Hans, and he says wildfire might reduce dynamo.
Yes, it might because it increases phosphorus. So that could have a suppressing effect on the dynamo.
Okay, interesting. Michelle then asks, just to clarify the last thing that you mentioned, was that dynamo did not greatly affect fish species within the Columbia Basin?
No, we saw no impact on fish condition or gut contents in the fish throughout the study. And we looked at a large number of rivers from a gradient of 0 dynamo cover to up to 80% dynamo cover. It did change the macroinvertebrate community that was present, but since there weren't food limited, they were able to get the food that they needed. So they more selectively fed on the species that they were interested in consuming.
Okay, so interesting. Okay, well, we don't have any more questions at this time, but I just wanted to say that at the end of our last talk with Brian Menounos, as I mentioned to this, there was this question of, what are the implications of a lot of what he had shared with regards to deglaciation in our region, and you definitely touched on some of them and helped expand our knowledge there. So thanks very much. I'll move into my thanks and a little bit of a conclusion. Thanks for bringing up that slide. So again, another round of thank yous to the Columbia Basin Trust for your support. A big thank you to everyone who's been donating to the series. We really appreciate your support there, and we appreciate you sharing the resource with your network. Again, if you are looking for more information on the series, you can find it on our website, cmiae.org. And a recording of this talk, along with the others, will be posted to the event webpage, as I noted earlier. Likely we'll get this recording up by Tuesday. And maybe somebody in our audience can put the link to the CREDtalks webpage in the chat right now, as opposed to our general homepage. That would be helpful. And then I'll just highlight that next week we're going to be welcoming Dr. Martin Carver with Aqua Environmental Associates. And Martin's going to run through some hydrological implications of what we've been learning about so far in the series. So as the hydrology for our region is one of the fundamental drivers in shaping the structure and function of our regional ecosystems, Martin's talk will help to further illustrate the implications of climate change for the Columbia Mountains region. So his talk is entitled Transition Hydrology en route to a new runoff regime. And it'll be the same time, same day next week. So we'll be meeting on Thursday, February 3rd at 12 p.m. Pacific. And I hope to see you all there. If you have any questions about the series, feel free to email myself. If you have any questions for Janice, she warmly welcomes you to connect with her as well. Thanks very much. Thanks, Janice.